This is a video about a monk called Gregor Mendel, and he is sometimes known as the father of genetics because he did some quite important work with inheritance of characteristics. The kind of living thing that he chose to do the inheritance experiments on were pea plants, and he chose pea plants because, and here's a extract from his writing, it says they may be cited the easy culture of these plants, so in other words they grow quite easily and they can grow in open ground and in pots, but also the relatively short period of growth. That meant that he could grow them very easily and they wouldn't take long to grow, so he could look at the results of his experiments in quite a short amount of time. And what he did, one of his experiments was to cross breed a tall plant with a short plant, and he was very careful that these were both pure breeding. In other words, if you cross two of these tall plants together, you would only ever get tall plants, and if you cross two of these short ones, you would only ever get short ones. And he crossed the two together to see what happened, and he found that the plants that he got were all tall. That he found interesting because in those days the idea that scientists had was that if you mixed a tall pea plant with a short one, or if you mixed the characteristics tall and short, you would get something in the middle, so you'd get something halfway across between the two. But he actually found out that that wasn't necessarily true. You either get one characteristic or the other. And to experiment further, he took two of these plants he had got from this generation. He called this the F1 generation. He took two of those plants and he bred them together. And interestingly, when he did that, he found that he got a whole bunch of tall plants, but also some short plants. He didn't find any plants that were middle or somewhere in the middle. They were either tall or they were short. And when he counted them up, he found, and again this is an extract from his results, experiment 7, he says, he found 787 that were tall and 277 that were short, and he gave that a ratio of 3 to 1. And it wasn't based just on this experiment. This is experiment 7, but he did a whole other load of other experiments as well, and he found that in circumstances like this he was getting a ratio of 3 to 1 in that second generation of plants. And he looked at this very carefully, and he came to the idea that the plants were passing on what he called inheritance factors. And remember, they had no idea about genes and genetics in those days, so he had this idea that he called inheritance factors, and he said that one plant would pass on one of the factors and another would pass on another, and depending on how they combined, you would get certain characteristics or features in the offspring. So this, we know, actually is the case and how it works, and the work we've done since then has actually backed this up. At the time, his work wasn't really recognized as hugely important, and we'll look at why that is in a minute. But what we can do is take this opportunity to actually do some more practice on genetic crosses and actually look at what happened in those pea plants. So if we're looking at crossing a tall pea plant with a short pea plant, we know that they were true breeding, so let's use capital T for tall and small t for short. Remember, they can only pass on one or other of these two alleles. So this one will pass on, in fact, can only pass on capital T or capital T. This one is small t, small t. And this is the pollen grains in the egg cell, so this is the gametes. And if we then combine this gamete with this one, we get this. And in fact, if you do all the possibilities, you'll only get these mixtures of alleles. Now you can see here, because we've got the tall in every single one, they're going to be all tall, and that's why back in this slide here he got all tall. They were all capital T, small t. He then crossed two of these together, and that would have been the following scenario. They were both tall, but we know that they have these alleles. And again, if we do the cross again, this time I'm going to use the Punnett square method. You can use either method in the exam, but I'm going to use the Punnett square method just to remind us of how that works. We'll take the alleles from this plant, so these are the gametes. They either have capital T or small t, and then this one down here. And if we do the cross, this is what we get: tall, 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 short. So you can see here we have 3 tall to 1 short. Okay, so these are the possibilities, all the possibilities that can happen, and you get 3 tall to 1 short, and that's why in his experiment he got a ratio of 2.84 to 1, which he rounded up as a 3 to 1 ratio. You don't exactly get 3 to 1 all the time in reality, because you have to remember this is due to chance, the crosses. So you have to do a lot of experiments to get close to the actual true ratio, but that's what's actually happening based on what we know today. The last thing I want to just quickly go over is why his work wasn't recognized at the time. There's 3 key points really. He wasn't actually a scientist, he was a monk. And so that might have reduced his credibility amongst the science population, but he also, or the science community, his work, when he did his work, he didn't actually publish it widely. I think he published it in like a gardening type magazine or something like that, or gardening paper. So it wasn't really published to a whole load of people. It wasn't published in a science journal, and at that time, as we said, genes had not been discovered, so people didn't really understand the idea of genes and how this could have worked. So for those 3 reasons, it wasn't actually recognized well at the time. But further experiments were done later, and about 40 years other people were able to reproduce this work, and they went back and saw what he had done and realized the importance of what he had done. Okay, so this is just basically an overview of Mendel and his work. What you really need to be able to do is just have an overview of the importance of his work and what he did, and apply it to some genetic crosses from what we know today, and maybe give 1 or 2 ideas of why his work wasn't actually recognized or discovered till long after his death.